power of scientific ideas, analysis of data, breakthroughs, understanding nature and the study of human needs. Here, the ideas of the East are supported by Western technology. Here, collaboration between the best minds creates new solutions. Here, each discovery makes our lives easier and the environment cleaner and safer. The International Science and Technology Center is a platform for science collaboration and the promotion of science. This film will show the results of ISTC's work in Yerevan, the capital of Armenia. After the collapse of the Soviet Union, when the Iron Curtain was drawn back, scholars in post-Soviet countries became free. However, at the same time, investment into science collapsed. Armenia, with its great scientific potential, was one of these countries. Saving science in the new reality became a critical mission for Armenians. Armenia was one of the most developed parts of the Soviet Union in terms of science. 33,000 scientists in the country with a population of just over 3 million is a high rate, and it was a great shock for Armenians to lose funding and support. Most scientists decided to leave the country. According to statistics, 80% of them left Armenia.
We were the most advanced country, our scientists were some of the best in the Soviet Union, and in one day we lost everything. We became independent, but we did not know what to do with this independence. There was no way to do science in this economic situation. At that time, any help was essential for us. That is why we highly appreciate what ISTC did and is still doing in Armenia. The financial help we received literally helped science to survive.
For many years, the Soviet Union had been considered a great power. Quite a few weapon organizations and scientists were located in Armenia. Armenia was one of the first countries in the post-Soviet region where ISTC opened a branch office in 1998. In an effort to provide work for weapons scientists, the International Science and Technology Center began to engage them into the peaceful projects.
Among all post-Soviet countries, Armenia demonstrated strong performance in programs related to space, nuclear power, nuclear physics, and microbiology. Most of these programs served USSR military needs. There was a risk that scientists who were involved in the Soviet projects could be converted by terrorists after the collapse of the Soviet Union. So the engagement programs run by ISTC helped us not only survive, but improve international security. As an institute, we were focused mostly on military needs during the Soviet time. After the Soviet Union collapsed, we were on the verge of shutting down. Most specialists were leaving science, and it was quite dangerous to lose people with such a rare knowledge and experience. And at that moment, help came from nowhere. We received grants, we could afford really expensive equipment, and our scientists started traveling and meeting colleagues from different countries. Nowadays we operate at full capacity and have great plans for the upcoming future. Weapons scientists started working in peaceful projects and learning how to use their experience and knowledge for civilian purposes. So as a result, we witnessed our transformation of weapons scientists into normal scientists who have the opportunity to work in collaboration with colleagues from around the world. And as a country, we received huge benefits. We stopped the brain drain, we transformed military knowledge into useful applications, and we obtained great results out of almost 200 projects funded by ISTC.
A new generation of scientists in Armenia. The problem of aging of scientific specialists in Armenia became pronounced since the early 90s, after the liquidation of the Soviet Union. A whole generation of talented researchers dropped out of science. The average age for a candidate of science has reached 50 years. Every sixth scientist is a pensioner. Half of the researchers are older than 50. Only 15% of the college students continue into science after graduation.
By the beginning of the new millennium, Armenian science became recognized as one of the oldest in the world. 40% of graduate students had left science due to the low income. The reasons were obvious: low wages, the absence of material base for research and development, bureaucratization of the organization of scientific work, and the low prestige of scientific work. Since the mid-2000s, the restoration of Armenian scientific specialists has been a government priority. Today, young specialists receive financial support through both domestic and foreign programs, through grants, foundations, and governmental organizations. And ISTC is one of the main supporters of the young scientists in Armenia. The situation has changed for the better since ISTC started working in Armenia. Nowadays, we see that more and more young people are getting involved in scientific projects. Usually, they start by doing a master's and PhD and then decide to work full-time. There are more opportunities in science for young people nowadays. They can travel, publish their papers, work in nice, fully equipped laboratories, and work in collaboration with the scientists from around the world. There is a demand for scientific work among young people. Students come to take internships and decide to stay. Participating in such international cooperation efforts helps them to get a better understanding of the current situation in the scientific world. We even encourage them not only be involved, but also take responsibility, manage the process or team. By doing this, they get experience, get trained, and becoming scientists of new era.
Tigran Dadlian is a young research officer. He is involved in the project on controlled light propagation in structured nonlinear media. Recently a student, Tigran is already taking responsibility of some parts of the project. By using photorefractive crystals, Tigran and his colleagues tried to study novel features and new possibilities for controlled light propagation and localization in the induced waveguiding structures.
The importance of the problem will be more understandable if we consider the optical beam as an encoded optical information carrier. Thus, the controlled optical beam propagation in waveguiding structures is equivalent to the controlled information transfer and addressing direction to different information transfer channels. Such devices are called all-optical, since both formation of structured media and information transfer are performing by optical laser methods. Important is also that the information carrier is the light or photons, elemental particles of light, as an object with most possible speed of propagation, which in turn provides the maximal speed of operation of all optical devices. Such devices are promising for optical storage and readout, addressing information, communication systems, and a new generation of optical computers. The main technical success of the first year of the project was the design and fabrication of an all-optical device which is based on the quadruple-like waveguiding structure in strontium, barium, iodide crystal, and demonstration of controlled light localization to any of waveguiding channels. The fabricated device operates as an all-optical photonic interconnect, providing an efficient and controlled addressing of optical information to any of 4 waveguiding channels. The fabricated interconnect can be a basic building block for more complex multi-channel interconnects. I believe that the start of the project is successful and results obtained during first year of the project are published in prestigious journal Optics Express. This is a good basis for further studies, and young scientists involved in the project received great experience. They work really hard in collaboration with our colleagues from around the world, and we admire their contribution and commitment to science.
International collaboration within ISTC. The nature of modern science as a global endeavor is increasing for a variety of reasons. Enabling factors such as advances in communication technology and cheaper travel have played a part, but the primary driver of most collaboration is individual scientists. In seeking to work with the best among their peers and to gain access to complementary resources, equipment, and knowledge, researchers fundamentally enhance the quality and improve the efficiency of their work. The revolutionary changes that occurred in Armenia in the 1990s created many problems for science, such as a dramatic decrease in funding, the decline of the scientist's social status, and a low demand for research from the economy. But since then, a lot has changed. Armenian science is no longer isolated from global science. One project on targeted elimination of multi-drug resistant salmonella brought together scientists from Armenia, Georgia, Tajikistan, Denmark, UK, Belgium, and Ireland. It is aimed at the identification of one of the most virulent and antibiotic-resistant salmonella strains and the development of novel phage therapy strategies to prevent future salmonella outbreaks.
Today, collaboration has never been more important, with human society facing a number of wide-ranging and interlinked medical challenges. The project started a year ago and already has made a number of significant accomplishments. The project teams have created collections of clinical strains isolated from patients with salmonellosis in their countries. In addition, the TAHI team has collected a set of salmonella strains isolated from food products to compare with clinical strains. All these strains underwent clinical, biochemical, and serological characterization and were tested in laboratories in Denmark, Armenia, and Ireland for susceptibility against antimicrobials. Based on the results, the strains with multi-drug resistant phenotypes were identified and selected for further, more detailed investigations. These experiments have revealed considerable differences in drug response depending on geographical region, which is an important finding of this part of research activities. Another important task within the project is the development of effective therapies of salmonellosis based on the application of phages. Here, we perform the screening of the existing salmonella phage collection to find the most efficient phages that kill the most virulent and drug-resistant salmonella strains. Comparison of phage susceptibility of the salmonella strains originating from Armenia, Georgia, and Tajikistan have shown some interesting results. We will proceed to genomics characterization of selected strains, and based on the overall results, we will create therapeutic phage cocktails that are most efficient at eliminating MDR salmonella strains.
Coordination of the work in a big team of participating institutions from 3 countries is a very important task for the successful promotion of the project, and all participants of the project actively communicated and discussed the project's progress in order to provide the efficient coordination of the project activities. The operative exchange of information on the progress in collection and testing of human non-typhoidal Salmonella enterica strains took place between the teams from Armenia, Georgia, and Tajikistan involved in the project. The work pages and progress of their implementation were extensively discussed during the workshops organized in last year within the frames of the project in Tbilisi and Yerevan.
Commercial potential of the project. Increasingly, the dialogue between science and business is becoming a two-way source of innovation. Many projects within the ISTC have commercial potential. One of them, microbial biogeography of the transnational fermented milk, Matsoni. Matsoni is a traditional national food product of Armenia. Little is known about the effects of biogeography and milk type on the microbial biodiversity of Matsoni or the fungal composition of Matsoni fermentations. High-throughput marker gene sequencing was used to survey the bacterial and fungal communities of Matsoni from different milk types and regions throughout Armenia. Results demonstrate that both production region influence Matsoni microbiota, suggesting that the traditional production methods preserve the transfer of unique regional microbiota from batch to batch. Investors pay huge interest to this project.
In Armenia, we believe that our research universities are powerful engines for economic development. Therefore, we try to focus on applied research. In this project, we study strains of lactic acid and bifidobacteria cultures potentially endowed with probiotic properties, in particular, capable of assimilating oligosaccharides of milk. The main goal of the project was to find and identify an effective complex of probiotic cultures of microorganisms along with oligosaccharides used as the only symbiotic drug for increasing the functionality of the human intestine.
The results of the research showed that traditional methods of obtaining fermented milk allow the preservation of unique regional features of microflora distribution. In this laboratory, we studied 180 samples of fermented raw, not pasteurized milk of domestic animals, such as buffalo, donkey, cow, goat, and sheep, that were selected and studied from different regions of Armenia, Georgia, and Nagorno-Karabakh, in more than 400 strains of rod-shaped and coccoid lactic acid bacteria and yeast.
At the same time, more than 130 samples of fecal masses of newborns were studied, of which more than 300 PCR products of genomic DNA were obtained. In bacterial profiles, as a rule, lactobacilli bacteria of generic origin Lactobacillus and Streptococcus predominated. At the same time, the species composition of the yeast sharply differs, the most important distinguishing feature of the region and milk type. The results of these studies made it possible to establish the regularity and specificity of the distribution of lactic acid, bifidobacteria, yeast, other microorganisms in fermented milk samples, and create a genetic map based on climatic and geographical regional features of origin. Dedicated and studied promising probiotic cultures of lactic acid and bifidobacteria can be recommended for the creation of new therapeutic and prophylactic and dietary soy milk products. The project was successfully completed within approved 24 months with funding of $210,000. The results of the project are reflected in a number of scientific publications published in prestigious foreign scientific publications.
ISTC has become a part of life for Armenian scientists. Every day they come to work and try to find new solutions, new ways of thinking, and move science forward at least one small step. It is how it should be and how it is.